All right. Well, I'm going to start us off here then. So today I'm going to be talking. We're going to be talking about Fusarium head blight today, just because it's one of those topics everybody's always interested in, and getting some updates in terms of management and some of the tools we have. That's what I'm going to be talking about, and then Dr. Rutkoski is going to be talking a little bit more about variety selection and resistance and what that can do for you in terms of managing Fusarium head blight. So, this is one of my favorite diseases to work on. I've been working on it since about 2013-ish. I really like working with wheat, and I've always really enjoyed working with this disease, even though it's destructive. So from a farmer's perspective, it's bad, but in terms of from a pathologist perspective, it's a very nice system to work with. So in terms of our situation the last couple of years with Fusarium head blight, we really haven't had too many issues, which in my mind has been a little bit surprising due to some of the weather that we've had right around flowering. So remember, it's that having that humid, wet weather a couple weeks just prior to flowering through flowering really tends to promote the development of Fusarium head blight. So in 2019, we had some issues with some of our, in terms of uniformity in the field, within fields as also as between fields, just due to that really weird winter and fall that we had, where it would warm up for 3-4 days there. It was getting up into the 60s and then it would freeze again. And so that was causing some issues in terms of uniformity. And that probably impacted the amount of disease we had, because it actually kind of hedged our bets a little bit. So everything wasn't necessarily flowering at the same time or similar times within or between fields. And didn't really have any issues in 2019. This past year, it was, obviously things were a little more uniform and looking better, but we also had a lot of wet weather right around flowering for many parts of the state. And when we looked at our models, it was predicting that, at least for susceptible varieties, that we were going to be in pretty high risk situations. But when you switched our model into that moderate susceptible or moderate resistant component, we weren't really at high levels. And I should have put some of those images in here because I had those, but I really think that was probably due to the kind of cooler temperatures that we had when it was wet. And we can still get Fusarium head blight when it's cool and wet. It's just we need a lot more rain. There needs to be many more days for us to really allow that fungus to, quote unquote, wake up, increase its growth, and increase the amount of spores that it produces, because those are what's going to be causing the infection. So I think that's probably why we kind of saw some isolated issues with Fusarium head blight this year. So we've been lucky that it hasn't been something that's really devastated us the past couple of years here, and that's a good thing. We don't need anything hurting our wheat crops more, and we're already dealing with low commodity prices and shrinking acreage and everything else. That's a good thing. So I like to just, want to start out by giving just a little bit of an overview of the disease, because a lot of times, we get this Fusarium disease confused with other Fusaria, because this is a fungus or a fungal genus that has a lot of members to it. And some of them are going to be more specific or cause issues in other crops. So Fusarium virguliforme is one that causes sudden death syndrome in soybeans. Fusarium graminearum is the one that predominantly is going to be causing Fusarium head blight in Illinois. Fusarium graminearum also causes stalk rots in corn, and its sexual phase is actually causes gibberella stalk rot and gibberella ear rot in corn as well. So this is a pathogen that has a pretty wide host range. It's what we call a non-obligate pathogen. So really what it does is it produces toxins that kill the tissues, and then it lives off that dead decaying tissue. That also means that it does very well growing on dead decaying tissue on your soil, so residue and things like that. And because it does have a wide host range, that means that as long as you have residue on your field, you're going to have the potential to have this inoculum produced. And I'll talk a little bit more about how it spreads and how far it can spread here in a second. But as with many diseases, this is a very episodic disease, more so than our foliar diseases, because it does have a very limited target window to cause disease. It's only going to be affecting our plants when they're flowering. And you do have to have those heads out and flowering to get those severe infections and then the production of vomitoxin. But when it does happen, when you do have those conditions that are going to favor significant epidemics, it can be very challenging to control for many different reasons. One, which Dr. Rutkoski is going to talk about, or Rutkoski, sorry, I'm not very good with names either, is that the resistance can be very, very difficult. It's not complete resistance or immunity in many cases. Also, wheat tends to flower over several days. So you have main tillers and secondary tillers, and that makes it challenging. And then usually when we're going out there and we want to make an application for head blight, we're trying to target those heads. And if it's raining for 5, 6, 7 days in a row, that can cause problems for us too. We could get in too early or too late. So a lot of different challenges for us with managing head blight just because of the way it works. In terms of the disease cycle, this is kind of graphically what we're looking at. So we start off, this is, like I said, a residue-borne disease. So if you have any kind of crop residue, but wheat and corn, it's going to be mostly corn that we're dealing with in Illinois, soybean as well. So it can grow on soybean, but it grows better on corn than soybean. When it gets wet and warm, typically is when you're going to start seeing that fungus wake up, come out of dormancy. It'll start to produce fruiting structures on that residue, and it produces 2 different types of spores. These very large asexual spores here that kind of look like boats, those are called macroconidia. And then it also can produce sexual spores, especially on corn residue, these little balls, and they produce ascospores within perithecia. And what's interesting about these is that these are going to be forcibly ejected. So it will shoot these out of the perithecia into the upper atmosphere. And that's really important for kind of this moderate to long-term spread of the disease. The macroconidia are larger. They don't move very far. These are the ones that tend to be locally dispersed and move from your residue up into the canopy with those hard driving rains right around flowering. So those spores need to land on a plant at the right growth stage. So it needs to land on flowering heads or heads that are just after flowering. That tends to be when we see the most severe infections. The fungus then can cause symptoms. So those symptoms are going to be usually bleaching of the heads at the point of infection and above. So depending on when you look at your heads, you might see individual spikelets or whole sections of spikelets that can go down with this disease. And that's because of how it infects. So if you have a flowering head here, and this is your spore, and it lands on the head and it's wet, it's going to grow into that spikelet. And if you think about the way a head is, it has that rachis right down the middle, right, where all those spikelets connect, and you've got your xylem and phloem there that's kind of feeding those with carbon and water. Well, the fungus grows inside that rachis at that spikelet point. And depending on the wheat variety and the environment, it can grow to varying degrees in that rachis and kind of clog up that tissue. And when it does that, it chokes off that water and nutrient movement. And that's why sometimes you do see these little sections where you say it infects here, everything above that will be bleached out and dead, because it's basically choking out that head. And then the fungus can continue to grow and infect, and as it grows and infects, it can kind of get into your grains there and produce those tombstones that were those kind of lightweight, chalky kernels, because there's a lot of fungal material in there. It's lightweight. And also sometimes it can produce vomitoxin or deoxynivalenol, or DON as we like to call it. And that's really important because, as I'll talk about in a minute or two, that's something that is not, we get docked for that if we're selling for human consumption. And also if levels are too high, we might not even be able to sell our grain for animal consumption. So typically what I tell people when I'm talking about managing head blight, I try to focus on the vomitoxin because that is something that we can't blow out of our combines. We can't clean it up really in our grain elevators, because a lot of times, not a lot of times, but sometimes we can have vomitoxin increase in our kernels and not necessarily have tombstones or symptomatic grains that can be sorted out. So this is what it looks like in the field. There are lots of different things that can cause bleaching, frost damage, there can be insect damage, there can even be root diseases that can cause this sort of head symptom. So with head blight, typically, especially following a good rain event, you get out there early in the morning, and you can see kind of this salmon colored, almost pink-like growth, typically at the base of the spikes. If you had a microscope and a little pin or a needle, and you could scrape that off, put that on a slide, and you'd see a whole bunch of those macroconidia growing right here on the spikelet. Mentioned the production of tombstones. So this is what tombstones would look like, usually kind of pinkish white in color, lightweight, shriveled grain. Usually these are going to be loaded with vomitoxin. We're talking about 2 or 3 figures here in terms of the amount of vomitoxin in a relative to healthy kernels, which are nice and plump. This is what we're shooting for, most of those healthy kernels. Now, as I mentioned, the ascospores can move aerially, so those are ejected forcibly and they move in the atmosphere. And some really interesting research coming out of Cornell actually showed that those ascospores will sit up there and they'll mix in the atmosphere, and then at night when conditions calm down, they actually deposit themselves from the atmosphere. So they shower themselves onto plants, onto fields. And this means that this is really it's not a local field to field issue, it's a regional issue. So as long as we're producing lots of corn, small grains, other things, there's just going to be a lot of potential inoculum that can move and still deposit itself on your field. That means that if you're tilling and you're rotating, yes, that can reduce the amount of disease because you might limit the amount of those macroconidia that can splash up onto your heads, but it's not going to be a major contributor in a severe epidemic or during an epidemic year. So some of the numbers being tossed out, they think roughly 70% of the inoculum that comes into most fields is being deposited from the atmosphere, which could be from distal sources, and then another 30% can be from within a field. Mycotoxins, as I mentioned, are very important. So the big one that we talk about is deoxynivalenol, although there are other types of toxins that can be produced by Fusarium, graminearum, and calmorum, and some of the other species that can cause head blight. These are measured in very small quantities, parts per million. And typically when we're trying to sell our grain, especially for human consumption, we're going to start seeing that dockage start at 2 parts per million, and sometimes even lower than that in some areas. But the important thing to keep in mind is that you can have elevated vomitoxin without those head blight symptoms, without those tombstones, and that's important because we can't clean those up and we can't take that out of the stream of grain. And so if that ends up in our grain stream, it ends up with, we end up with lower profits for everybody because then that has to be blended down in order to sell that grain. So this is an example of some of the levels that we need to be able to sell our grain. So as you can see, if we want to sell it for human consumption, we're trying to get under 1 part per million. When I think about managing head blight, my main goal is during an episodic year, a really bad year, I want to be able to at least have my producers be able to sell their grain for animal consumption and get something back for it. We don't want to have so much vomitoxin in our grain that it ends up being basically radioactive and we have to just dump it out and lose that grain and not get any profit from it at all or any money back for it at all. So that's usually how I approach managing head blight. Nothing we do can guarantee that you're going to get under that 1 part per million for human consumption, but the idea is we're going to be basically think of managing head blight as a pizza. We want to get rid of all that pizza, which would be the head blight, So we're trying to take out a couple slices at a time. So hopefully that we don't have very much to deal with at the end. So I'm not going to get too much into integrated management because I want to talk a little bit more about some of the research that we're doing and update you on that. But if we think about managing head blight, it is something that has to be managed using an integrated strategy. If you're just going out there and you're using one tool, you're not going to be able to build a house, okay? You're going to have your saws, your nails, your hammers, all that stuff together in order to put up your house. So think about managing head blight that way. You want to assemble it and use all the different tools you have in your tool belt. So the most important thing for managing head blight is variety selection, making sure that you select a good moderately resistant variety that meets your production needs, and you can do that in various ways. I'm sorry I misspelled your name. I like that W in there because I have a couple in my name, I think. But anyway, that's something that she's going to be talking about a little bit later. As I mentioned earlier, residue management is something that you can consider. Trying to promote residue decomposition is kind of what I tell people. Sizing that residue, breaking up into small pieces if you're not into tillage, just making sure that it's decomposing as fast as possible. The other thing some people like to do is plant after soybeans. That is because soybeans are a host, but again, the Fusarium graminearum fungus just does not grow as quickly and does not produce as much inoculum on soybeans. And so from a local level, again, that might reduce some of your potential macroconidia that can splash up onto your plants. But don't expect that to prevent your head blight, but it can definitely help. Then the thing that a lot of people are familiar with is applying an appropriate fungicide at the right timing. So if we think about adding these up together, a good variety will get you a 40–45% reduction in visual symptoms, another 35–40% reduction in vomitoxin. Residue management might knock off another 5%, and then your fungicide about 45%. Reduction in vomitoxin and 45 to 50% in those visual symptoms, those scabby kernels and things like that. And it has to be applied at the right time, and you have to use the right product. And then the last thing you got to do is you got to be paying attention to your fields and knowing which fields you have that are showing symptoms of head scab. Getting out there if there is a significant amount of the disease and trying to do what you can to separate your good fields from your bad ones. Sometimes in severe instances, you might increase the fan speed by 20–30% to try to blow out those scabby kernels, and making sure you get that wheat out of your fields as fast as possible, because the longer it sits there, the more fluctuating wet-dry periods, the more vomitoxin that you can get. And then if you can store it, we want to be storing this, making sure it's nice and dry below 15% or so. I like to tell people the drier we get, the closer to 13, the better it's going to be, because that fungus will not continue to grow and reproduce in the bins if that moisture is below 18%. If it continues to be wet, it can continue to grow and produce vomitoxin in those bins, in addition to other nasties that can grow in your bins. So in terms of some of the research we've been doing, we've been looking at some newer products here that have been coming out in recent years. So we've got new fungicides, chemistries that are being released or will soon be released. These have a succinate dehydrogenase inhibitor mode of action. That's basically something that's going to inhibit the ability of the fungus to produce energy for itself. The one that everyone's probably most familiar with right now is Miravis Ace. In the next year or two, there's going to be a couple other products coming out from other industry partners that will also have this sort of mode of action as well. This is different from the fungicides we typically use to manage head blight, which are demethylation inhibitors that are going to be impacting the ability of those cell membranes to basically have structure and fluidity. Those are the Carumbas, the Prosaros, Proline. Those are our standards that we've been using now for at least 10 years, I'd say, for managing head blight or more. So What we want to know is we want to know if these newer products, Miravis Ace and some of these newer products have different or wider application windows, because that's something that some people have been talking about, and also it's a different mode of action, so it could have some different properties as well. And we also want to know if these products work as well as some of our standards like the Carumbas and Prusaros for Fusarium head blight suppression. So what we've been doing the last 2 years or more actually, but this is the project we've been focusing on the last 2 years in Illinois, is assessing the ability of Carumba, Prusaro, and Miravis Ace applied at 3 different timings, either at the 50% heading. So when those heads are about 50% of those main tillers have started to emerge, or are about 50% emerged from that last ligule there. That early timing, that's when some people are interested in. Flowering, that Feeks 1051, and then 5 days after flowering. Research has shown, again, research we've done through this kind of coordinated trials, that the best efficacy for fungicide application for head scab is between that flowering to that 5-6 days post-flowering window. Just a little more information too. We always like to compare these combinations here with our varieties, and then we have untreated checks as well. And then we measure visual reduction in Fusarium head blight on the heads. We also look at the percent of Fusarium damaged kernels, yield components, and then vomitoxin. Unfortunately, the vomitoxin data take a while to get in, so I don't have those to present. And as I was saying, those are the most important data really when it comes to this, but we can get an idea of what we're working with and how effective these products are. All right, and just keep in mind, these are preliminary analyses, and I'm only going to show you statistically significant data. So that means that I am confident that these differences are more or less real, that they're just not due to chance. So if we look at the effect of variety here, moderately susceptible, moderately resistant, on the percentage of those heads that have Fusarium head scab here, you're looking at, I don't know what this is, probably about a 30% reduction or so in just by going from moderately resistant to, or moderately susceptible to moderately resistant, both in terms of the disease severity and the percentage of Fusarium damaged kernels. So this just shows you here the benefit that we saw just by getting a good moderately resistant variety in 2019 relative to a moderately susceptible one. In 2020, it was a similar story. Again, numbers are a little different. And keep in mind, we're doing these at 2 different locations. So these are averaged across location. So we are seeing, again, you know, a reduction in the amount of visual symptoms, as well as, you know, a good 30% or so reduction in the percentage of Fusarium damaged kernels that we see as well. So that the use of a moderately resistant variety, I like that the best, because you don't have to worry about timing. You don't have to worry about your growth stage. It's something that's always just an inherent property of your plant. So you can kind of count on a moderately resistant variety giving you that benefit than if you would have went with a moderately susceptible one. And that's why I like to really talk about that. And the other thing, back in the day when we first started looking at some of these resistant or moderately resistant varieties, they had these big yield drags on them. And we're not really seeing that with a lot of these newer varieties. So here in 2019, there was no difference in yield, even though there was a difference in head blight. And in 2020, our moderately resistant one actually did better than our moderately susceptible one. So if you get yourself a good moderately resistant variety that fits into your program, that can be a real help for you with managing Fusarium head blight. In terms of our fungicides, again, these are the fungicides that we were utilizing at the rates for Fusarium head blight suppression on the labels. These are non-treated checks here. Keep in mind, no interaction between product and timing. That means that no particular product did better or worse at any timing in 2019 than another. So they all behaved relatively similarly. So these bars here are averaged across all those timings. That's probably why they're a little elevated, because we're including those earlier timings as well. But if we just kind of look at the relative response of these fungicides. Our Miravis Ace did a little bit better for us with the visual symptoms here relative to the other ones we tested. And then when we looked at the percent damaged kernels, Miravis Ace and Carumba kind of stood out here with that. Fusarium, again, did well, just not quite as well as those other two in 2019. In 2020, we didn't have any differences in Fusarium damaged kernels, but again, the same kind of pattern here we're seeing with our products with the visual symptoms of Fusarium head scab. So the plants look like less of the heads are damaged. Again, that Miravis Ace doing a little bit better than our Carumba and our Fusarium in this case. When we look at timing in 2019, this is what we saw. Again, regardless of product, that 10-3 timing was our worst timing. So we had a roughly 13% severity in 2019, I believe. So there was a little reduction. Didn't matter what product we used, but they all kind of, didn't do as well as those timings between the flowering and the 5 days post-flowering timings. In 2020, we saw that again, that 10-3 timing was not the best for us and relative to the flowering timing, which really gave us our optimal control in 2020 across our different sites. So we're still going to be saying that flowering timing, we have this little window at the back end, which is going to probably give us the best suppression of disease. It's probably better to go in a little later than in going in early. Sure, you can get some reduction, but it's not going to be nearly as optimal as it would be if you get in right at that flowering timing. The other thing that people ask oftentimes with these fungicides is, well, what about these other foliar diseases I have? Staganospora leaf blotch is probably one that we encounter most commonly because it's residue borne and it's really something that does kind of have a local effect. And so what you can see here is we were rating in 2020 on our fields, our flag leaves that were affected at soft dough. And you can see here relative to our non-treated checks that all of these products did a good job at reducing Staganospora leaf blotch, with Carumba and Miravis Ace doing better than Fusarium in this case. But they all have a good effect on that. They also work well on rusts and some of those other things that kind of might move in later in the season. Yield effects, again, that timing, in addition to reducing our head blight the most, that timing, the flowering timing to 5 days after, were also our best treatments in terms of yield. And that's timing. And our fungicides, when we look at our fungicides, all of our fungicides increased or improved our yield relative to that non-treated check, with the Miravis Ace and Carumba doing a little bit better for us than our Fusarium in this case. In 2020, yields were the greatest in Miravis Ace relative to Carumba and Fusarium, but again, all of those products improved our yields relative to that non-treated check. We also were looking at the effect of rate. So that's something people have questions about, whether or not increasing the rate can improve the reduction in Fusarium head scab at the different timings. So what we did is we went out with those 3 products again at different timings. So at that heading, flowering 5 days after, and then we went in at different rates. So in the case of Fusarium, you have the low rate, which is 6.5, and the high at 8.2. And then Carumba, you have that 13.5 and 17 rate. And really, here's our non-treated check. If we look at our severity, what we're seeing here is that, with the timing, again, is really important in reducing these diseases for all of them, but that rate really wasn't giving us much of a benefit in terms of reducing the amount of Fusarium head scab across the different timings for Fusarium and Carumba. That you can go in with the low rate and you're pretty much going to work out at a similar level as you would at the high rates. And that's kind of holding true again with some of the yield data as well that you can see here. But again, always doing better than our non-treated check for the majority of these treatments. One other item to note, sometimes people will want to go out and make a treatment for a foliar disease. Maybe you get stripe rust coming in early. Maybe you've got some really bad Staganospora starting up, or it's just been pounding rain early. Going out and applying a strobilurin fungicide is something that's been demonstrated now, and this is again a publication that came out at the end of 2018 in Plant Disease. This is a citation, but basically what we've shown is that going out in a field and applying a strobilurin only product, which would be something like Approach or Headline or Quadris or the generics with azoxystrobin, that those actually can increase the amount of vomitoxin relative to a non-treated check. Going in after making an application of one of those QOI or strobilurin products with one of those recommended DMI fungicides will reduce the amount of vomitoxin, but it won't reduce it to the level that you would get just by if you applied that DMI fungicide alone. So you do have to take some caution if you do want to come in earlier in the season and make an application. I tell people you should at least be considering some sort of mixed mode of action fungicide to try to minimize the effect, because a lot of our fungicides for foliar disease suppression will have a QOI in them. The other thing you can do is just wait and hope that that later timing is going to be efficacious in managing your diseases. And I think in many times that is the case. So with that, I'm going to finish up my talk here. Might have went a little bit long, sorry about that, but keep in mind variety selection is going to be your heavy hitter for head blight management. There's lots of new varieties out there, so learn how to select them and go with good ones that you can find. Keep in mind, we're still looking at the vomitoxin data. So just because we're looking at visual differences here in scabby kernels and scabby heads, again, that doesn't tell us much about the vomitoxin. So I really want to make sure I have a complete story in the spring to share with everybody and see what the effects of these timings and rates and things like that actually have on the vomitoxin. Still focus on that flowering timing to 5 days after timing. Try not to go in early. If you have to make an application, going a little late usually is better than going a little early. And I say that just simply due to the nature of flowering in wheat. Again, the primary tillers and secondary tillers, that whole flowering process can take a week, 11 days, 12 days or longer, depending on the weather. So you always have some heads that will be covered going in a little later. And just keep in mind that you will get suppression of your common diseases on the foliage with a lot of with our products that we currently have for head blight. And with that, I'll just wrap up and say, thanks today. Thanks for attending, and everybody who's kind of helped out with this work in the last couple of years. And I'll take any questions if we have time for them. Otherwise, I can wait, or you can email me questions, whatever y'all want to do. What do you think, Nate?
Does anybody have any specific questions or comment before we switch gears a little bit and hear from Jessica? You're welcome to unmute yourself, or you can type them in the chat box. We can take some more questions at the end, but certainly if there's something that's on your mind specific to what we just mentioned, you're welcome to ask now. I tell you what, why don't you go ahead and we'll go ahead and get this slide switched over. And if you want to stop sharing, and Jessica, you're welcome to share your screen.
All righty, all yours.
Hello, can everyone see my presentation?
Yes.
Okay, great. Yeah, so I'm Dr. Jessica Rutkoski. I'm the small greens breeder at University of Illinois. And yeah, my presentation is just going to be focusing in on wheat scab resistance in Illinois. Well, scab is a tricky disease because it's due a lot to environment as well as genetics. For instance, just different characteristics about how you manage things on your farm, the crop rotation, the tillage, the temperature, the moisture, and then there's a lot of random factors that we don't necessarily understand, which also determine whether we're going to get scab or not. So it's up to a lot of these different environmental factors that are known and unknown. And then also genetics does play a role. And we see, time and time again that essentially the scab that you see in the field is really due to 30% genetics and 70% environment. And that's why, management is a really important component. But the genetics is really important as well, because you can really stack the deck in your favor by planting a resistant variety or a moderately resistant variety. With that said, because of this environmental component, if you plant a moderately resistant variety, that's still not a guarantee that you're going to have a really, really good control, because that year could end up being a really, really bad year for scab, and you'd still get pretty bad disease. But, I think over, in an average year, you're probably going to be doing, pretty good job at controlling, using a moderately resistant variety, and especially if you're going to be applying fungicide. Another tricky thing about scab resistance is it's based on many genes. There's been a lot of work done to try to understand the genetics of this resistance, which has amounted to more than 500 different scab resistance genes that have been reported. And it sort of starts to look like a complex trait similar to yield or other agronomic traits, which we really don't have a single gene or a silver bullet that's going to reliably provide adequate levels of resistance. There are some large effect QTL, like for instance FHB1, which you might have heard about, and this one tends to have a little bit larger effect than many of the other ones, but it's not something that you can. It's not diagnostic, so you won't be able to say this one gene is going to give me resistance or not. It doesn't work that way. There's an interaction with that gene and all the other genes in that variety, as well as interaction with the environment. So it's just really important that keep that in mind, that there isn't like a single gene that you can look for in your variety that's going to give you resistance. So what breeders have been doing, especially, over the past 20, 25 years, is just breeding as we would any other quantitative trait like yield, just using selective breeding and making incremental progress over time. And what this has amounted to is actually really, really good sustained incremental progress in resistance. So this, I did a study of the different wheat breeding programs, improvements in Fusarium resistance, and this is in the northern part of the eastern soft red wheat growing region. And I found that there's been about a 1.3 reduction in Fusarium damaged kernels per year, and a 1.4 reduction in vomitoxin, 1.4% reduction in vomitoxin per year. So that's really encouraging. That means, over time, every year, our varieties are getting better and better and more resistant. And that kind of raises the question, can wheat breeders actually eliminate scab? And I actually think the answer is yes. I mean, if the current trends continue in variety resistance, based on these trends that we're seeing, and if we continue to put effort and resources into improving resistance to this disease, I estimate by the year 2096, scab will no longer be a significant problem in our region. So that would mean almost every variety available should have really good resistance by that time. And scab would be no longer a problem. So that's kind of the vision for the future. And I think it's possible. That's a long ways away from now. So I'll just kind of come back to the present and talk a little bit about the resistance among currently available varieties. So before I get into that, I'll just briefly talk about kind of what goes into this evaluation of scab resistance. So we put everything into a misted and inoculated nursery that really creates a very conducive environment for high levels of disease. Then we do some visual scoring on the plots also harvest those plots and do some scoring and analysis on the grains. We send grain samples off for DON testing to determine how much vomitoxin is in the grain, and then take all that data and do data analysis and combining data also from not just the current year but previous years if it's available. So this year, for the Illinois State Variety Trial, I went through this process. I also used data from last year, data from this year, and I was able to come up with a pretty robust resistant category classification of the varieties. I'm showing here on this graph, this is like the distribution, a histogram of the ratings that I was able to give based on all the data that we've collected. And those ratings were based on spike symptoms, kernel damage, and vomitoxin. So the vomitoxin is weighted a lot more in that score, but the other spike symptoms and kernel damage also were included because we know that these are, if they're weighted appropriately, these can be very predictive of vomitoxin. So in some cases, we didn't have the vomitoxin data because it takes a year to get it. And so by using these other traits, I can get a pretty good prediction of how, that level of resistance even for vomitoxin. So I got this distribution. It's a pretty normal distribution as we would expect for a trait like this, and just split it into thirds. So the most resistant third of the varieties I classify as moderately resistant. The middle third are moderately susceptible. So on average, the varieties are moderately susceptible. And then the worst third are the susceptible category. Now, because this is a tricky disease and, there's a lot of other factors like environmental factors that play into the actual expression of disease, there is error associated with all these ratings and all this data. So in order to kind of account for this, try to account for this error as much as possible, anything that I couldn't neatly classify into these categories, I gave them more than one classification. So, and this is just because there is some margin of error. So something got a rating that is kind of near the threshold and it has some error associated with it. It's not, I can't say with a high level of confidence that it fits into either category. So if you look at the Illinois State Variety Testing report on wheat varieties, you'll see these scores given. So that you'll see varieties that are moderately resistant. You'll see some that have a score of MR/MS. That means it could be either moderately resistant or moderately susceptible, but I can't say with a high level of confidence that it fits into either category. And then there are varieties also with the MS/S, which means it's either moderately resistant or moderately susceptible. And I can't say with a high level of confidence either way. And then there are some which are very clearly susceptible that I can say, yes, I'm 95% confident these are susceptible. So I just want to highlight the moderately resistant varieties that I can, and these are the ones I say, yes, 95% confident that these are moderately resistant. And if I were to repeat this same experiment again, I would get the same results. So I just want to show you a little bit what the data looks like. So when if you go to the variety testing website, you can get the full list and kind of go through it and look for yourself and be able to interpret it. So I have the variety name here. If you go to the variety testing website, it also says, the company is a separate column. So you can, more easily identify the source of the seed. And then I have the score, which is on a 1 to 9 scale, with 1 being the most resistant and 9 being the most susceptible. And then there's a plus or minus here that shows you kind of the margin of error. So how much, this isn't an exact score. It's a score that has, there's a little bit of wiggle room there because there is noise and error in the data just due to the nature of the disease. Then I have the resistance category listed. pretty straightforward. It's either, moderately resistant slash moderately susceptible, moderately susceptible slash susceptible, and susceptible. So those are the categories. And then I have a column here to show you how many years of data was included into this scoring. So obviously more years of data is going to give you a more robust result, and versus just 1 year of data. Let's see. Yeah, you can find the complete results if you go to the University of Illinois Variety Testing website. And it's just available at this link here. And you're going to want to click on this file or this link that says 2020 wheat circular PDF. And there you're going to find not only the scab resistance results, but also results for yield across multiple locations. So you can take that into account when selecting the varieties. Also, I think I forgot to mention the first two here are, these are checks. So these are our moderately resistant checks used in experiment. And there's also a susceptible check there. If you go and you can also look and compare scores with those checks to kind of get a sense for how they compare with some of our known varieties that we really know to be moderately resistant. And I just want to, just like what Nathan said, some of these varieties are also really high yielding. So you don't necessarily have to make a choice between a high yielding variety and a moderately resistant variety to scab. You can probably find a variety that is good in both of those categories. And so that's all I had planned to talk about. I just want to acknowledge a few people contributed to this work. So Fred Kolb, my predecessor, and then 2 research specialists, Olivia and Elias Handel. One of my colleagues, Juan Arvelais, and several student workers who really do a lot of help and contribute a lot into the phenotyping. So if you have questions, you can ask them now, or you can always email me at jrut@illinois.edu. Does anyone have questions right away?
I did go ahead and put that link in your instructions to get to those ratings in the chat box. So if anyone wants to, can click on that link and see that. Does anybody have any questions? You're welcome. If you want, you're welcome to unmute yourself. You should be able to click the unmute button in the lower left corner, or you're welcome to type something in. I guess, Nathan, I do have one question for you. I know in our area we did have some issues with stripe rust. Is there any, I guess, any at least brief thoughts on manage on stripe rust that, especially looking into to next year, that guys should be thinking about?
Well, with stripe rust, again, that is something that typically is going to be blowing in from warmer regions every year. And so we do have, if those of you that are familiar with the monitoring system that egg pest monitor system that we're using for tar spot and southern rust and corn and soybean rust in soybean, obviously. Those platforms also are being used for stripe rust as well. So we're hoping to get more participation next year, but that's one way you can kind of trace stripe rust and where it's moving and showing up. Because if it arrives early, before heading, that's obviously something that can really affect you during a cool, wet season. And it has a very short life cycle, which means it produces a lot of spores very quickly. So if you have a susceptible variety, you're going to want to get out there and make a decision. If it, probably have to make a fungicide application if it's shown up early. But if you're already getting close to heading and you are already planning on making an application for head scab, well then, you don't have to worry about an additional application. Plus, if you're somebody who doesn't want to spray anything, you know, that again, that's something that can definitely, when we've had trials where it takes out 40 bushels, no problem. So it can be really bad on a susceptible variety in a bad year. So I would say keep track of those websites and keep track of the newspapers and Twitters and blogs and whatever else. And if you start hearing about stripe rust showing up early in the season, you know, think about what you might want to do there and how your fields are looking, their yield potential, and if it's something that you're going to have to maybe add additional management practices during the field season.
Would you happen to have that, the website for that tracking that you could maybe put in the chat, or have that be able to maybe grab that and throw that in for people?
Yeah, let me see if I can find it.
Okay, nope, I appreciate it. So certainly, if you have any questions, I certainly, first I want to thank both Nathan and Jessica for hopping on. Hopefully this has been, you know, giving you guys some information just as we're getting, if you have any final decisions, especially on wheat varieties, we wanted to talk, mention that, and try to bring out some of the research I know that's going on campus out to you guys, and may give you some things to consider. I know we think about this a lot come maybe, you know, March, late February, March, we think about planning, but there's obviously a lot of things we can do now that that can help out too. So all right, we did have a question. If I need more wheat acreage, is it better to use corn ground or double crop bean ground that had that was wheat last year? Nathan or Jessica, have any thoughts on, especially with disease-wise, what would be a better option, corn versus double crop ground that did have wheat?
Well, I guess my thought would be that your double crop soybeans would be better than going into corn, simply because a lot of that small grain residue has already had a season to start breaking down. And then you're going to, the majority of the residue you're going to have on the field is going to be soybean residue. So you shouldn't have as much issues with head scab. Then the one thing you do want to keep in mind if you do that, if there still is some straw on the ground, is just to you know, make sure that you're not putting in a very susceptible variety in terms of Staganospora, because Staganospora is going to be overwintering on straw. So you want to make sure you have something that's pretty strong on the leaf blotch complexes.
And would it be fair to say that certainly if you had a lot of issues with disease, or certainly head scab, that would in your wheat that those double crops are in, that would be probably more problematic than if you had pretty good disease management in your wheat that spring before?
Right.
Anyone else have any questions? Well, I want to thank you guys for joining. This is meant to just kind of be kind of a lunchtime chance for people just to hear about a topic and certainly kind of discuss and share more. We are going to go ahead and do another one of these in 2 weeks. And Sarah, if you want to switch to that, we're going to do one. We're going to talk about cover crops. Actually, I'm going to talk about that, and just kind of want this to be kind of an open discussion. We can bring up some topics, and I'll hit on especially any planting decisions, seeding rates, and just things that we need to know here as the crop starts to come off, and we make some of those decisions, what we want to put in, and if we're interested in cover crops. So with that, I don't have any other questions. Thank you again, Nathan and Jessica, for for joining us, and we thank all of our participants too. So have a great day.
See you later, everyone.
Bye, everybody. Thank you.